Let's just starting with the brief introduction. My name is Faisal. I am senior scientist at Karolinska Institute in Stockholm, Sweden, and this is we welcome to all of you to this APPNA Merit Health Care Research and Innovation in Pakistan webinar series, which is a great effort, and it is really promoting a lot of new ideas and concepts and research in Pakistan. So this is a joint effort of APPNA Merit Central Committee and the Research Subcommittee. And today we have a pleasure to listen Dr. Faisal Khan. He leads the NCBC Precision Medicine Lab, which is an interdisciplinary and translational biology research group that is interested in developing and using genomic and multi-omic approaches to target cancer and the infectious diseases. Dr. Khan's lab started in 2013 and is now affiliated with multiple institutions in Peshawar and is funded by private and the public funds, including government of KPK and also the Federal Planning Commission. Dr. Khan recently appointed as the additional project director and inaugural assistant professor of bioengineering at the University of Engineering and Applied Sciences, Swat, and he is also the head of the Center for the Genomics and Sciences. He has won several awards, both in academia and also in the industry. He also advises to the federal and provincial governments on numerous boards and committees in educational sciences. Dr. Khan graduated with master's and the doctoral degree from University of Oxford and recently completed his training in cancer biology and therapeutics from Harvard Medical School. In 2019, he was nominated as a young global leader from Pakistan by the World Economic Forum. Very impressive, Dr. Khan. And it's a great pleasure, and we are very much excited to listen about the precision medicine and its challenges and everything, what you are going to do in Pakistan. So please share your slides, and so this will be 45 minutes talk, followed by questions and answers.
Thank you, G. It's an absolute honor to be here. Big thank you to Saeed Uzair Saab, who's made the introduction and who really kind of introduced me to the world of APPNA and Merit. And I really appreciate that, and I'm very grateful. I have quite a few slides actually, but let's probably dive straight into it. So is my screen up by now?
Yes, yes, please.
Cool. So I'm going to share what we've been doing here in Peshawar in Pakistan from a research perspective, which is very biased towards translation into the clinic. Our mantra kind of is that we've had enough of papers, and now we're really, really hungry for prototypes and solutions which could help us in our area in terms of medicine, help us with patient outcomes. And the key for that, we believe, is interdisciplinarity and a lot of entrepreneurial or innovative approaches that needs to be kind of flourished in the lab. So this is basically a snapshot of our journey so far, and along with some fresh technical results of the lab, for those of you who might be interested. So I know this slide's pretty wordy, but it's like half my disclosure, and I kind of wanted to put it out there. So I'm associated with all these organizations in different capacities with financial and non-financial interests. So today, I want to quickly kind of, for those of us who are away from the lab, quickly kind of warm up in what our main areas are. So my training has been in systems biology, which kind of underlies a lot of synthetic biology, and I'm going to explain what that means. And the goal here is to hatch something in Pakistan which could really help us specifically with patient outcomes, but broadly in terms of our economy. then some fresh vignettes from the lab, and I just probably end with some slides about life at the lab and some ideas on how we're circumventing the problems we face in research in Pakistan, which we wholeheartedly accept and try to hack our way through. So we'll start off with a quick molecular biology 101. We all know this complex, probably the most complex machine on Earth and the most intelligent one as well, hatches or assembles itself out of the first cell, which is a zygote. If we go inside a cell, there's a nucleus, which is packed with a set of chromosomes. If we pick up and uncoil a chromosome, which is very gracefully, sort of intricately, and we're learning about the 3D structure of chromosomes these days, so there's a lot into it. So these coiled structures, if we uncoil them and we arrive at the final unit, we see thread-like structures, which takes us to the very familiar double helical structure of DNA. If we use a magnifying glass and read through the double helical structure, we see a code, an ordered set of chemicals, four chemicals, which really kind of captures the complexity of life, which is fascinating. Matt Ridley, the famous author, in his book says, if I read to you the whole genome at the rate of 1 word per second, 8 hours a day, it would take me a century. So again, biology through the genomic period kind of quickly turned into a data field, a data science field, and we learn why. And this is one sort of shot at it. A single cell would have about 3.2 billion bases, which in genomics terms, if you kind of pick up all the raw data sets, it's about 240 GBs in a single cell, which is fascinating again. More scientifically, and I just browsed through the NCBI GenBank statistics just now for this talk, the growth in the number of unique sequences in terms of bases is right in front of you. It's an exponential growth in the amount of publicly available data. And as you can see, the slide probably made this in March 17, but if you look it up now, it's gone from 240 billion bases to about 1 trillion bases, which tells us about the enormity of the data sets that are out there waiting to be analyzed and sifted through. So again, it's a massive field emerging out of single cells, thanks to genomics and all the technologies. And the surge, as you can see in this plot, is right about the time when the first human genome was unraveled and published. We saw Bill Clinton and Tony Blair kind of announce that at the White House. Craig Venter was alongside, and the story goes on. So basically, a living organism is now a text file, like literally. This is a screen grab from my MacBook. For those of you familiar, it's a very common text file. This is Mycoplasma genitalium. This is the entire genome. Obviously, the first sort of field view screen grab. You have to scroll down, of course. But if you can look at the properties of that file, you can see it's just 0.5 megabyte. So again, the smallest bacterial genome, 0.5 megabyte, not that much. But again, the sheer availability of it on your desktop or laptop machine is just mind-boggling. And now we have the number of whole genomes available publicly are surpassing the graph I showed you earlier. So again, we're beginning to read the entire genomes of organisms as we go along time, and the field is growing really, really fast. For those of you who are not familiar, the genomic revolution has hatched several other what we call omic technologies, which are giving us several other omic data sets. By ohm, we mean whatever that molecule is, we pick up its availability in the entirety of the cell. So a whole cell data set, a pan cell data set, would be called an ohm, but then we have the epigenome. If you look at the mRNAs, we have the transcriptome. If we pick up the proteins, we look at the proteome. So the multi-omic approach now is sort of a post-genomic stage of biology, where we're trying to add up and integrate all these data sets to make sense of it, to derive biological and clinical meaning out of that data. So this is kind of at the core of what we do. This is where my training was, and this is where most of the lab is kind of focused on, and we love dabbling into all these data sets. The second piece is synthetic biology. If you've got a text file, if the data is so readily available, you can certainly now use computer-aided design to engineer life or living cells. And we all must have heard about synthetic biology in the way bioengineering is carried out these days. Just as examples, computer science allows you to code and build software. Electrical engineering allows you to sketch out different circuits and probably fabricate a chip at the end. What synthetic biology does is it's allowing you to come up with intelligent logic gates and beyond, things which are interactive and not just one-way expression of genes like we saw in genetic engineering. So we're able to design these complex systems, print the DNA out, and express them in different host cells for whatever application we're after. So again, the design, build, and test cycle like we have in engineering has now been adopted in biology. And this really, really fascinates us, me personally. I've always been kind of passionate about biology since my childhood, but I've always kind of been envious about engineers as to how they kind of spot a problem, they're able to forward engineer something to solve that problem. And in biology, we've hit that sweet spot now, and we're able to design, build, and test solutions, which is extremely fascinating and takes us to the world of biomanufacturing in a whole different manner. And we're really keen on hatching something around this in Pakistan. But I'll stop there. Just to give you an idea about the costs, the first GM product back in the days using traditional genetic engineering, the Golden Rice project was about 100 million worth of R&D dollars. If you do the same experiment now, it probably costs you less than $10,000. So that's again an exponential decrease in cost of R&D in the realm of biomanufacturing, more specifically the design and engineering of living cells through synthetic biology. So gone are the days of traditional biotech, and we're on to now synthetic biology, which is a way more, arguably more accurate and highly scalable way of looking into whole cells and engineering them. And over here, we're deeply passionate about harnessing this technology for Pakistan. I'll finish with this section with this example. I'm not into American football. I don't even know who this player is. But again, I usually use this slide as a quiz, but the necktie here is extremely fascinating. It's a product of Bolt Threads, I believe a startup based out of San Fran. And the necktie is made up of spider silk, but not your routine spider silk, which we sometimes touch at home somewhere. These are spider silk fibers which are brewed in yeast. And again, a classic example of how SynBio can enable us to reach into the consumer sector now, and consumer products are being tested out in the market already. So again, since we're a bio-based economy and the world is shifting towards more renewable sources of all these materials, we're again very fascinated about how we can leverage this in the Pakistani context. So systems biology, multiomes on one side, which will be the main topic of today, but we're also fascinated about synthetic biology, and I'm going to leave SynBio over there for now, but happy to answer questions down the line. So what is the Precision Medicine Lab? We're basically a federally funded precision medicine pilot project funded by the Planning Commission in Islamabad via the Higher Education Commission. And what we mean by precision medicine is, I'm borrowing the NIH definition over here, which is the use of information around genes, the environment, and the lifestyle of a person to practice medicine. Again, this is something a bit more futuristic, but the idea is to come up with more tailored sort of approaches to treating different diseases, in our case, cancer. Our argument in Pakistan was the survival rate in the country is almost half of the global survival rate, which is extremely painful. Another statistic was a fifth of patients die because of the chemo, the side effects of the chemotherapies that we prescribe in the country. Again, a startling statistic. And the amount of dollars saved or pain avoided is also humongous. And that's the argument we made at the Planning Commission. And we said, we really need to pilot something out around genomics and help us kind of begin to transition towards that. So there are 4 pieces of the pipeline here. We needed to have a biorepository. Then we needed to have a way to read those cancer genomes, exomes in our case, for now. And then how we could leverage that genome of our cohort with other omic data sets and see if we could do some systems medicine approaches and try and identify new drugs or potentially repurpose existing drugs. And then, of course, there has to be a way to do the preclinical analysis or the wet lab analysis and do those functional assays in the lab to validate what we find. So there are technically four pieces to the project we had 4 years ago, and I'm very proud to report we've assembled and put together expertise and infrastructure in all 4 of these. So the repository we have is an ultra low freezer, but the painful side was developing the governance structure, the SOPs around biobanking, and following all the global best practices. And here specifically, I'd cite the NCI guidelines for storing biological samples. We also had to go through the IRB approval processes, but we aim to, and with time, we've kind of upgraded not just ours, but also the surrounding sort of structures that we have, the institutional structures, and we've evolved very rapidly over the past 4 years. And we're very finicky about the details here, the documentation, and the informed consenting process. We sit in Peshawar, so we have this in 4 languages, English, Urdu, Pashto, and Persian. Then obviously there was the cancer genomic piece. The genomic piece, normally extracting the DNA is pretty straightforward. We need to have had the infrastructure to read out the DNA, but then the tough job is the analysis. What we do with the raw data. Over here, I'm just sharing a screen grab of CBio portal. This is the TP53 gene, human TP53, and the little circles you see are the mutations and the frequency of these mutations across the length of the gene. So it starts over here, it ends over there, and you see even individual genes have, if not thousands, hundreds of different mutations at different frequencies. And all of this noise signal has to be found within all of this noise, which is where the challenge starts to begin. We had the liberty to choose a cancer. So after a lot of consultations, we chose oral cancer. It was very obvious to think back now because it's dubbed as poor man's cancer, less than 20% survival rate. Most oftenly, like many others, reported in late stages. And it has a very high incidence and prevalence in Pakistan and broadly South Asia. Weirdly, probably not surprisingly, not a lot of data sets available out there. So we thought this could be a very interesting problem. This is a Globocan graph where we've plotted mortality versus incidence. And you see Pakistan somewhere at the top, which is pretty sad, to be honest. And this is kind of convinced us even more that probably this is it, and we really want to nail this. we want to dissect and go inside and see what are the main genetic players, and ideally, as we go along, what are the mechanisms of how this cancer evolves and obviously begins with in the first place. So the genomic layer is there, has to be done, and we're progressing towards it. COVID has slowed us down, we can concede, but we're going there. And the cohort, all of the exomes, we take normal samples and tumor samples. So these are pairs of samples from each patient, and we'd be getting the exome data as we hit our milestones. But we don't stop there. So we're lucky to be here at RMI. RMI is the largest private hospital in KP, and we're very lucky to have found them as a very digital savvy hospital. So they've got their EMR sorted, they have their radiology all digital, and we're helping them transition their pathology into the digital world. That gives us additional layers of data about our cohort, which becomes fascinating. But we're not going to stop there. We're already sampling for additional layers of omic data sets. And now, as you put layers on top of each other, becomes a candy shop for any data science research group. And we're calling it a multi-omic stack, which is literally a playground for machine learning and deep learning experiments. So we've already assembled stacks based on existing publicly available data, but obviously from white Caucasian populations. But we're extremely keen on seeing how the Pakistani cohort and its stack would look like. And we're hoping to have that in the middle of next year, inshallah. So we don't stop there, of course. The last piece is functional assays and screens. And the good thing is that we keep some aliquots of our biopsies, the tumor biopsies, and we grow them into cell lines. So we're beginning to have a nice little library of primary cell lines out of Pakistani and some Afghani patients who are seen here at the hospitals. And this becomes a very sort of fascinating sort of tangent in the research that we're doing. Once you have a library, you can test out all sorts of drugs, and you can do a whole new set of experiments using these cell lines. And since they're growing, obviously with their pros and cons as a 2D model, patient-derived cancer model, but again, with whatever we have, we can still grow them further and kind of scale up the library, replenish the library as we move on. So let's move on to some fresh vignettes for those who might be interested. As an example from Multiomes, Laiba, who's an MS student at the lab, she looked into the miRNA species, and she was keen on sifting through existing data sets of head and neck cancers and seeing what she sees as differentially expressed across different regions, stages of head and neck cancer. So we begin with a heat map of this sort and volcano plots, which help us sift through genes which are significantly differentially expressed. So as you can see at the top, these are genes which are differentially expressed at the early stages, and these are the sets of genes which are expressed at the late stages. Ideally, a biomarker should be expressing early on, so you don't miss out on the initiation of cancer. We also looked into the expressions of these guys using different methods. Again, different validations come across, and we narrowed down on 5 different miRNA species, which we are now taking into the lab to see how they matter in our cell lines, in the Pakistani and Afghani cell lines. We also studied them across different subsites, and these are some of the results. I might not have time to go into it, but happy to answer questions. Again, how the 5 species express differentially across different sites of the head and neck region. And these are some interesting survival analyses of the patients based on existing data and the specific mRNA species that we're looking at. And again, the two arms are separated very neatly here, which kind of gets us more excited about these 5 mRNA species which LIBA has spotted. And these are just some downstream enrichment analyses which help us decipher more about these mRNA species and their neighboring other human proteins encoded by genes to see where they kind of are located from a gene ontology perspective. And this work was recently presented at the Shaukat Khanum Cancer Symposium this year. Second example I want to show is how we are using these integrated multi-omic approaches to answer the question of why the human papilloma virus is more of a concern in oropharyngeal cancer and not that much in oral cancer. So Arsalan, who's a stellar undergraduate student, he picked this up with the help of others, of course, and he began by hunting down genes which were interacting with the human papilloma virus in the 2 different cancers. And what we use is our network topology, tools around network topologies. So basically we're looking at literally a Facebook of proteins within a cell. And the approach we use is guilt by association. So if you've got friends who are involved in cancer, you're more probable to be involved in cancer yourself. And that's the approach we use to decipher what our candidate proteins are doing in these networks. We also look into the broader neighborhood. So not the first neighbors, but also the second neighbors. So we kind of expand the network. And what Arsalan found through a cluster analysis using M-code was a cluster that you can see in blue over here, which was more about OSCC, oral squamous cell carcinomas. Again, with candidates who partially are already known, but 2 new candidates which need to be discussed further, validated further, and that's what we plan to take into the lab now. Arsalan and another teammate, Yusra, are also looking into the Ebv virus, which also plays a role in head and neck cancers. And we're trying to see an overlap between HPV's roles and also EBVs, which makes a fascinating sort of problem space. So again, another example of how we're sifting through thousands of genes using integrative multi-omic approaches to narrow down candidates that we can pick on in the lab.
Dr. Faisal, can you kindly use the slideshow, because some audience, they cannot see your slides properly. If you
So I'm using the slideshow.
Okay.
Is there a different way of doing it? I'm sorry.
No, actually, I think I can see the slides. They are projected okay. I don't know whether some others are not seeing it. No, your slides are, Doctor.
Should I move on?
Yeah, please go ahead then.
Apologies to them then, I'm sorry. I don't know what's happening. The second example is from the imaging group, which is looking at the image analysis group, which is looking at digital pathology. So obviously, a lot of biopsies are seen in the histopathology department, but we lack all the resources, especially all the fancy gadgets to scan these images. So over here, we went on an ambitious sort of project to see whether we can use low cost, what we can easily call screen grabs of the normal microscope and see if we can make accurate predictions, whether something's cancer or not, and also perhaps staging if it's a cancer. And Ebrar took on this project in the team along with other mates. And we've generated a local data set of labeled images. So we sit closely with the histopathology department. A big thanks to Professor Iqbal, who's been super supportive in all of this. And they've invested a lot of time into labeling these images with us at different magnifications. And we've captured those images using the conventional binocular microscope with a 5 megapixel camera. And the recent sort of improvement was crossing around 80% accuracy in the CNN models, the deep learning models that we're developing here. And this is remarkable for a screen grab, for a field view image of a conventional microscope. Normally, what you do is you get a gigabyte worth of a large scanned image of the entire slide, and that's what people use. And for that, then downstream, the problem is you don't need to. You also need to like pay. There's a lot of capital goes into the machine itself, but then you need a lot of compute power to analyze those heavy images and arrive at something. So over here, we're trying to circumvent that and see if we can use these normal screen grabs to predict cancers. Third one around signals, I'd like to share a wearable study that we've started. And we were aiming for 50 plus, the 50 plus females in our society, colloquially हम कह रहे थे कि अम्मियों वाला group who are normally on their way to some sort of CVDs, who are non-compliant, especially with their diet, etc. And we thought, can we use wearables to kind of track their details and see if we can predict any onset of serious health conditions? So we started off with a pilot within the team. We recruited 6 people, and we used a very low cost, like $30 off the shelf smartwatch, along with 2 more layers of data sets. One was vitals, of course, and then some other self-reported details about their health. And we integrated those, and we are now trying to measure which features kind of matter the most in what we're trying to accomplish. We've also been, one of our collaborators at the Aga Khan, Dr. Zainab, she recently kind of diverted our attention to pregnant mothers to see if we can track them in that stressful condition that they go through over the 9 months. So that's another interesting problem that we're trying to approach using these wearables. So we got some very interesting data, raw data, which we're still analyzing from the wearables from a 30-day data set. We recently presented that at the ISMB conference, which is the flagship annual conference of the International Society of Computational Biology, and also the European Society. And Hamad's talk also won him a prize here, again, validating how important these data sets are in the medical space. And if you can't afford an Apple Watch, there's no need to shy away from whatever's out there. And that's sort of the hungry approach we're using here to glean all the data that we can. My final example here is going to be EMRs. Again, this credit goes to the hospital here for maintaining their data in a way that we could analyze it. But again, when COVID hit us, like everyone else, we tried to repurpose and see how we could help out using our skill sets and expertise. And we went through a lot of analyses, and there was a molecule which one of the team pointed out from existing data, from the data which is coming out last year. And we spotted a molecule which was of interest. I can't name it since the team is already thinking about a startup around that, but let's call it the molecule X. So what we did was we approached RMI and their data team and asked if we could have, and the hypothesis was that this molecule would be overexpressed in the urine of those patients, of COVID positive patients. So we sifted through all the COVID positive patients who'd taken urine exams, and we got the data out of the database here at RMI, and we realized there was a difference that we hypothesized based on the data which was out there. So it's a very common molecule, and it's kind of overexpressing underexpressing in COVID patients. And this is what we're picking up. So Abdus Salam was the guy who did this. They're already trying to dabble with the name COVID-Actor, perhaps, maybe something else. But we're trying to approach this using a proper clinical study now as we move on. But again, a classic example of once you have interesting, high quality data around you, and if you come across a nice question, you're only a few hours or a few days at a maximum away from your early details and early results. And this is what really keeps us going, all these different types of data sets used in a concerted manner to answer the questions that we have. Let me have a sneak peek of the chat window if there are any questions. If not, I'll move on. But I don't seem to be able to look at the chat window.
Go ahead with this, and then we will take up the questions by the end, 15 minutes
okay.
before. Yeah.
Okay. So, so what is the lab and life at the lab? I'll spend the last few minutes on this one. So we're a unique, this was all possible because of consortium. I was based out of CCOS University here, and we collaborated with Rahman Medical Institute, the hospital, and again, a really a nice sort of personification in a way of a great academic industry collaboration. And it's also a public-private partnership in a way, because the main chunk of money came from the HEC, but I'm also extremely grateful to the two institutions who also co-invested in cash into the project, which we normally do not see. And I really hope this becomes an example for others in the country. So again, the lab is supported by a consortium of highly committed organizations. So CCOS is one of the oldest engineering universities in KP, and RMI is one of the largest hospitals in KP. And we're truly privileged to be working with both, and also, of course, powered by the Higher Education Commission through the National Center for Big Data and Cloud Computing. So we're 30 odd people at any given time, with a clear bias towards prototypes and solutions and some papers on the way. But we really want to, I personally really want our young people to taste sort of the glory in a way of coming up with a solution which touches a patient. You know, when your prototype for the first time functions, or your code runs for the first time and it works, I really want our youngsters to kind of feel that sense of accomplishment, that moment really. And I don't think they can look back after that. So hence a clear bias towards prototypes. We started off as a new lab from scratch, so I had to wear a thekedar wala hat as well, doing all the engineering, helping out RMI's engineering department. And again, they're very generous of having donated an entire floor for us, which we kind of redid from scratch. And again, a hint for people who plan to do R&D here, you're not gonna get a ready-made lab or a bench space at an established institution. You'll most probably be having to develop it from scratch. This is what we had 3 or 4 years ago in its brand new sort of state. It's 4 years old now, and much busier. We also began to get some compute power, which we're very excited about. It's not much, but it kind of does our job for now. And we also call it the little beast. So we've got Abrar and Hamad playing with their images right there. This is just part of the team, but I want to give a big shout out to all the collaborators, the co-PIs. I don't think we would have been doing anything without our clinical investigators or partners, Dr. Iqbal, Dr. Mushtaq, Dr. Zubair Durrani, who's been extremely passionate. Most of these are oral and maxillofacial surgeons who are giving away the tumors. Dr. Ahmad Khan's recently joined the rank and in principle agreed, and we're hashing out the partnership with his hospital in Mardan now. Fazil Razik Saab is the pathologist, and Johar Saab, our co-PI, was part of the Human Genome Project back in Washington, back in the days. So again, privileged to be surrounded by all these senior and junior people who are way more smarter than I am or we are. We're broken into 4 different teams for now, just to bring some order to what we do. But mind you, we also have other exciting avenues which I'm not able to talk about today, including astrobiology, biology in space. So I was recently put on a NASA program, a NASA Star 2 program for space bioscience. And again, we learn a lot from there. And we also use our tools to dabble into data from the International Space Station, data around the stress which astronauts kind of face, and also the biological experiments. But I'm happy to touch on any of these in the Q&A. Again, all these partners in these hospitals, I'm not going to name them individually, but a massive sort of gratitude to all of these guys. So what's life at the lab? I have my last 4 minutes here. And how are we learning to circumvent all the problems? You know, when you come across anyone in Pakistan, or most people, or people who've kind of decided to live abroad, they normally cite all the problems, but there are always ways to circumvent them. And we'd like to stay positive and high-spirited here in the lab. And part of my job is to insulate our lab from all what's happening around us. But how are we learning to circumvent? Here are some examples. We're totally mission-driven, and we work hard on this. This is something you do actively. This is not a passive mission. So we came up with this mission statement together in the lab, and that's what we live up to every single day, to glean every single data point available to optimize cancer treatment and minimize ineffective treatment, to help every cancer patient and their household. Meeting the patient and their household here in Peshawar, in the situation they're in, really moves every single one of us and keeps us going. We also do value sprints, just to remind everyone in the lab of what our values are. And these are not my words, these are their words in answer to these 3 questions. And you can get a feel of how everyone's thinking against the status quo, if I can say that. We also have an elaborate HR program, so you can't really hire the normal way. We have a 4-tiered program. It's very painful. We're kind of proud, but at the same time sad to say we only have 1% acceptance. 1% is huge, and it really makes it difficult for us to turn down all the exciting talent that's out there. But the 4th layer of the selection is the selection conference, where everyone comes, the finalists, they come spend a day with us, full of activities and some tests, of course, to measure their teamwork and also their depth, the depth of their expertise, which they claim. And trust me, people who don't even get selected, we get thank you emails from the experience they get. And I think this is our way of giving back as well, giving back a taste of what actual R&D looks like or should look like. It's real life, really real, because it's not just a lab. We're in the middle of a hospital. We work very closely with the OT. During COVID, we sampled some of the severely ill patients, and we're working on the genetics of that. And it was grueling. It was very sort of taxing, to say the least, on the team. But again, a great motivator for us to come back to the lab and do the job we're doing and contribute. It's cross-disciplinary, so it has its pros and cons. You have to go out of your comfort zone. But again, the boundaries are where all the magic happens. It's independent. We work very independently, unlike many other labs in the country. We try and give liberty to the people. We trust them with their intelligence, but we hold them accountable in different meetings and measure their outcomes. It's constant feedback, institutionalized. We have different ways of, we have different steps of orientation, midterm reviews, and self-assessments. And that's pretty much it. Other than that, everyone's free to do whatever they want, but they have to report on these 2 occasions. When we look after the people, I think we try and help them excel in their careers. As an example, the last 5 levers we had are all down here, and Asma, to begin with, is in Europe now, using an Erasmus scholarship Habiba is in New York using a Fulbright scholarship. Mariam just left us to Plymouth on a fully funded scholarship. And Junaid and Ahmar are going to leave us for Poland and France on fully funded positions. So again, we try our best to kind of help facilitate them. These are just some cross-disciplinary trainings or sprints, we call them, in the lab. So every skill set is passed on to people who are interested, and we make sure this happens regularly under a schedule in the lab. And you can see the variety of the topics there. We also have external seminars to bring in more flavor, more ideas. We also have high school internships. We have some stellar kids through a highly competitive program. There's a guy from Carrot College Cohort, and this guy who's in Imperial College now. He came from Atchison College. So again, trying to give them a taste of R&D in Pakistan. We also have a hack series around us, a hackathon series. These are 5 startups which came out of it. 2 of them have raised money now. They're trying to validate their products, and the journey goes on. So we're really keen on kicking out some products out of the lab as well, not just R&D. And it's always fun. We have a common room. We have meeting rooms to help them, give them space. We have an annual symposium where people dump their ideas. Alumni swing by and give talks. You see Fahim here, a visiting student from Vienna, who's a founder now of a company in Vienna, and all the other goodies that we have in the lab. And we make sure everyone has a good time. So we go on annual retreats as well, as you can see here up north, which is always very exciting. So I begin my acknowledgements here to all these bright young minds who really, really inspire me every single day in the lab. And I'm really grateful for their decision to join the lab and help contribute into the work we do. I also want to say a big thanks to HEC. We are a private institution, and normally people don't really trust, the public sector does not really trust, I dare to say, private institutions with research funds. And I'm so proud that they trusted us, and I hope they're happy now on the amount of stuff we're delivering, and we're beginning to deliver more on that. So a big shout out to everyone at the HEC and the NCBC. And I want to close with these guys. Rahman Saab, founder of Rahman Medical Institute, top class cardiologist in the country, and engineer Tanveer Saab, who's the founder of CCOS University, for not just giving us all the things they've done in kind and the moral support, but also the investment they've made in cash, in our dreams, in our research, trusting us with our vision to do science. And I really, really hope others kind of follow suit and name some labs after their families and their loved ones for perpetuity, inshallah. Please visit our website and our social media channels. And I'm gonna close again with these institutions. There is a lot happening, and we just shared what's happening at Precision Medicine Lab. I'd love to speak on any of these. Please join us on our journey towards world-class science here in Pakistan. And I thank you all for your patience and this great opportunity to mingle with you guys.
Thank you very much, Dr. Faisal. Very fascinating and completely new vision that we are seeing actually in Pakistan. Dr. Swat, would you like to start with some?
Yes, of course. Well, Dr. Faisal, honestly, I'm speechless at the amount and the type and the quality of work, and your leadership, your articulation, command on English, and your philosophy of dealing with people. And it looks as if you are, one person with, I tell you, about 100 attributes that are amazing, and the amount of work you're doing is just outstanding. And you're I'm speechless. The type of work you're doing and bringing up the younger people. My only concern is that that the momentum that you have maintained with your with your personal attributes and your expertise, I wish and pray that these momentum can be carried to in your lab, as well as there is emulation of what you're doing in other places. And the type of work you do, honestly, we did not know that it is being done in Pakistan. And we would, I'm sure many of us will be happy to visit your lab. And I'm not going to take too much time because I'm sure there are many other people who are doing so. My best possible compliments to your work, Doctor.
Thank you, G. Appreciate that. You're very generous with your words, but I'm glad you liked it.
Great. Now, Dr. Muhammad Ahmed, he's also joining us very soon. But yeah, I think this is overall very wonderful, and my impression is also same that. Do you think that this is too big for there, and you can really there are so many projects and so many different visions within this whole setup, and you have enough resources and workforce and everything? That is a little bit the concern, that exactly what Dr. Swat has also already pointed out. But would you like to comment on this, Dr. Faisal?
Sure. That's a difficult question, but my quick answer would be I'd like to remain focused on the resources we have and make the best use of that. Obviously, I have to kind of wear that fundraising hat every time my funds begin to run out. That's my job. But part of my job is also to insulate my team out of from these sort of challenges or constraints, if I might say. It is challenging, but I don't think we have any option but to pull this off. The uncertainties that we face downstream shouldn't stop us from expanding or going deeper into the things we love.
Hidden structure.
Dr. Muhammad Akbar Akhtar Hamid has joined us now. Welcome, Dr. Saab. Do you have any comments to or questions to Dr. Faisal?
No, I'm not at the moment. Please carry on.
Yeah. So taking this opportunity, so I have so many questions. It's very exciting overall. Dr. Faisal, what you're doing, and this is completely new as in Pakistan. And I would like to know, how are you dealing with the ethics or ethical issues? Because here you are dealing with the data coming from patients, right? And then you have a private hospital which is engaged with the government sector. So my question is, who owns the data by the end, and where it is stored? And do you have any safety features to protect the data of the patients?
So we have an elaborate storage and anonymization process of the data sets that we have. Happy to share details on any occasion. And you're right, it's challenging. Like, people are not used to elaborate paperwork or documentation or ethical procedures, which sometimes actually slows you down. For example, the amount of time we spend with the family before the surgery, for their consent, the patient and the family, finding out which language they speak first, and then taking a team which is appropriate, which has a clinician as well, which has someone who's literate in the project. It takes a lot of time, but make sure we do it. And credit goes to the team and the patients. They have to be doing this every time, every single time we get a sample. So it was very hard to begin with. Even the team was not convinced. I'm sure some of them who are listening now must have been saying, Dr. Saab, kya kar rahe hain, kyun tang kar rahe hain itna zyada? But I'm sure everyone realizes now that this was important, and I'm glad we have this set now, and we're off to a good start, I believe.
Brilliant, brilliant. So I was just going through some of the recent data on the mouth cancers, and we know, the typical studies, or like a lot of studies, they show us that it is mostly linked with smoking and like alcohol consumption or human papilloma virus, or the chewing of the beetle quid like pan, right? But there is a recent study, I think a couple of studies, one came from India and one from Taiwan, where they have actually found a very strong link with the polluted air in the population. I don't know if you're aware of this or not, but this was published in Journal of Investigative Medicine, and they found a very strong high rise of the oral cancers in those areas where there was a strong air pollution. I think your study, the facilities that you have and the workforce and the technology that you have, it would be of great interest to look at this angle as well, because then we will know exactly that in cities where we have very high rate of air pollution, from diesel particles or from petrol or other chemicals emitting from the industries in those surrounding areas, what is the incidence of the oral cancer? Do you have something on this to say?
So we're aware of secondhand smoke or passive smoking, tobacco, of course, but we haven't been aware of urban air pollution as a risk factor for oral cancer. We are aware of the Indian group who are doing a lot of good work, but we are already kind of invested into tobacco and its smoked and smokeless use, and also secondhand smoke. We were also surprised to find how mechanical stress, like broken teeth, a broken tooth could be a risk factor in oral cancer. And we're working on that as well on the molecular side of it to see how inflammation kind of correlates with the data sets that we have. But that's an interesting study you cited.
Any question from Dr. Sarwat or Dr. Muhammad Akhtar? So then maybe perhaps we can also take some questions from the audience. And there was, I think you have already answered this. The precision medicine lab provides any opportunity to the students for internship. And I think you have already discussed this. That's highly competitive. Definitely people are welcome to contact you, but it's like, as you said, it's 1% acceptance, something like that. And
we're always happy to receive emails or through our jobs page on the website.
And then there's one question from Nusrat Ali, is any correlation of oral cancer with the use of tobacco, like or Naswar, commonly used in KPK? Have you looked at this, and what is the incidence?
So we're obviously recording what our patients have been up to lifestyle-wise, and it's already established that Naswar is the menace in the north, and Pan and Gutka is the menace in the south, and we really need to counter these two in our country.
I have a question. This is Sarwat Hussain, Dr. Sarwat. Dr. Sab, do you do contract work also? If, for example, somebody wants to have, very simple example, if wants to have certain chat bots or health bots, for example.
So we're looking at both now. We started exploring opportunities on the data and the preclinical or the cell culture model side in terms of contracts. But we'd be happy to discuss opportunities, and I'd love any one of you to point us to any of these.
Because sorry, go ahead, doctor.
No, continue, please.
No, please go ahead. I've taken too much time. Go ahead, please.
Okay, so just one quick question. First of all, my apologies because of the time zone difference. I missed part of your talk. I'm sitting in Canada, but I wanted to know about your view on the increased rise of marijuana and their products and how they're affecting our oral mucosa, because this is something very unofficially used, especially the north part of it. And the Canada being the first country as a whole, not the first country, maybe second country as a whole to allow the marijuana to be used. You can buy it even with license or even without it with the shops after providing certain proof. How do you see this thing in Pakistan, especially, well, my understanding as a pediatrician, that marijuana has more effect on the cognitive part, and also some effect of the children who is before born, they have some subtle changes, but not a huge side effects like other serious drugs, like something in the heart or so. So how do you see relate this thing, which is very commonly used in a different products?
I wouldn't be able to comment on that because I haven't come across any literature which indicates marijuana or its metabolites as a risk factor. I'd be keen to see how many patients have disclosed use of marijuana or related products. Normally, people shy away from disclosing even Naswar. We've had a grandma who had a tumor, but she reluctantly disclosed she's been using Naswar for 20 years. But not every patient is kind of comfortable disclosing. But I'm very keen to see now that you've pointed marijuana, but I'm afraid I wouldn't have much information about its role in oral cancer.
Thank you. Then we have a question from Dr. Zafar, please. Zafar Sab, can you please ask a question?
Hello.
Yes, Zafar Sab, please kindly ask your question. Yes.
Are you hearing me? I'm Professor
Yes,
Dr. Zafar.
yes. Yes.
Okay, thank you very much for the speakers' very elaborate talk on this precision medicine and precision lab. I, as a physician, would just be tempted to ask that whether the precision lab can be helpful in identifying the epigenetic changes which may occur in people with unhealthy lifestyle, overeating, and propensity to develop diabetes and obesity, because these sort of changes do bring chromatin changes in the genes leading to metabolic processes. The second question of mine will be that epigenetic changes due to hyperglycemia and methylation of DNA are responsible for the oncoming long-standing complications of diabetes, like endothelial damage, nerve damage. So could the precision lab, Faisal Saab, at your institution may be helpful if we can, identify such patient who may go and at least sort of counsel them that they should bring proper glycemic control in their disease. Thank you very much.
So I didn't get the second part, but I believe you're into the molecular mechanism of what causes that. So yes, I think we'd love to explore problems like these, which are of local relevance. We do have a couple of surgeons and consultants who've come to us with their sort of trend that they saw, and we begin to dabble into data using their perspective or hypotheses in mind. The first question, epigenetics, absolutely. That's something we have expertise in, and we've been looking into DNA methylation data over the past couple of years. We're also storing our enough sample to be able to look at the methylation patterns of our cohort, oral cancer cohort, if provided with enough funds to do that experiment. It's just a matter of running those samples through a machine, and then we'll be able to decipher what comes out of the raw data. But yes, we have that in mind, and we have the expertise to look into other cases that anyone might be interested in.
Perfect. Dr. Shahid, Rafiq, I think you have some questions. Dr. Shahid? Yes, please.
Can you hear me?
Yes, please.
Yes, my question is that it was mentioned about the private-public partnership. So we are aware of, in other countries and in North America about the concept, and it's Pakistan government, especially the new government, is quite focused and encouraging towards that direction. But what is your experience so far, or vision, or your gut feeling as to how much is the private part is protected by the state government or the public portion when they decide to take over the whole project.
Sorry, what do you mean by decide to take over the whole project?
I mean, so one fine morning, if the government says this is our project and that's it, or you can take these peanuts to the private entrepreneurs who have been investing in that project. So isn't it that the private-public partnership, that private entrepreneurs are investing in a project where the land or the building is from government, and or so the new government comes and say, no, this project is not good because it was started by the previous government, and let's fold it or let's restart with new partners. And what happens to the old partners?
So I'd actually be worried about the problem the other way around. I think the government lacks the capability to protect its interests in areas such as ours, in terms of data or samples or IP. I think the government is usually more worried and concerned on whether we should even fund something that we cannot protect. So it's the other way around. I've seen projects fail between the government and the private sector. But most of the times my observation is that the governments tend not to undo projects, they just tend to move on, which is again a bad thing because they need to reflect and look into the outcomes of whatever they invested in. So normally the water keeps flowing and there's no looking back, which has its pros and cons. But you can hardwire your interests, and the government can hardwire its interests during the initial conception of the project. I don't know how familiar you are with the Pakistani system, but there's this, the actual project note is called the PC1 thing, the PC1 document. And that's like taken as a Bible in terms of implementation and execution. So it's all down to how nicely you write that PC1 document and how you kind of preserve the interests of each stakeholder. And I've hardly seen governments sway away from the PC1. Yeah. So that would be my observation.
I think this is the Dr. Faisal. This is the same question, kind of like that I was asking you about who, by the end, who owns the data, right? For example, I mean, if there is a patent coming out of it, and since this is a public-private partnership, then is there any contracts between state or HEC who is sponsoring your activities? How do you, I mean, so far you have not encountered any problems, but is there anything on documentation that's probably, I think Dr. Shahid wanted to.
So we're lucky our project PC1 has been very liberal. It secures our rights. It encourages innovation and entrepreneurship, which is commercialization. It only asks for a level of effort put into commercialization. It doesn't even ask for any revenue or any sort of claims in there. It just asks us to make sure we try and commercialize and set examples for projects to come. That's the very positive side of it. In terms of data and protection and whether it's public or not, we've been allowed a 2-year embargo. So the moment we get our data sets, we are allowed to have a 2-year embargo, and after that we'll have to make it public. And generally in our field, you do make it public by submitting to the NIH's GenBank in order to get your paper somewhere. You can't get your paper without submitting your data in these journals, right? But if there's anything IPable before that, patentable, I think that has to be taken care of during the 2-year period. So, and we've been lucky because, again, it's well thought out. The PC1 was well thought out, nicely written, and B, it was liberal enough to give us the incentive to run with whatever we come up with and make sure we use it in the best interest of our people.
Excellent. So Dr. Syed Uzair has recently joined us, sir. Welcome.
I want to,
Do you have any
yeah,
questions?
I want to add, yeah, I just want to add a positive
Yeah.
note here, and that is we need to kind of empathize that like ethical standards or clinical trials or commercialization and all the questions around IP, we don't have precedents in Pakistan. Like we don't have great precedents or enough numbers of precedents to set the system right. So if I usually go with an open heart and have that empathy and think to myself every morning that we've never done this, so let's help them learn as well. And if I use that approach, I've hardly faced anything adversarial or counterintuitive in a way on my journey so far in my 8 years. I think with a little empathy, we'll realize that since it's never happened and it's happening for the first time, and it kind of brings some responsibility as well to be able to be very honest and fair and be able to chart out a win-win situation and not be selfish about things in a very bad way. I think we are in the process of laying the foundations of a healthy research culture here. So it comes along with a lot of responsibility.
Just one quick question, which I think one of the participant has also requested. How have you managed to publish your work, particularly this in related to this, and how and where did you publish? If you can share some info.
Yeah, so, there was another problem. So writing is not our forte. English is our third and fourth languages. So publishing has been, and we don't have postdocs in the lab. We only have one recently. So there's no culture of someone helping the PI. So we've been slow in publishing, but we are gradually coming up with stuff. We've had dozens of conference papers in different conferences. We can share those. We also have some fresh preprints out on medRxiv and bioRxiv. You can look them up. And we have about a dozen manuscripts on our way now that our things have matured. So we concede it's been very, very slow, but it had its own reasons. And we have a good lineup of papers coming out, which we're very excited about.
Thank you.
I have a question, actually a comment, Dr. Faisal. I'm in Virginia, Richmond, Virginia, and a professor of radiology here. And I actually run a journal myself as a chief. I started that like 5-6 years ago as a journal of global radiology. And as a result, have some resources available personally and otherwise who can help you with writing the paper and take away the difficulties with with the speed with which you can finalize the paper, especially scientific writing. So and of course it will obviously be done ethically and confidentially. Be very honored and happy to help if that's what you require.
Thank you so much. I'd love to speak to someone on that. And it's a very difficult position that I get put into because you need things out soon, and but you don't want to compromise the quality of the stuff you put out in your name and your team's name. There are shortcuts which we get tempted with, but we're resisting it so far. So let's put it that way.
Brilliant. Any further comments or questions from panelists or the participants? Dr. Uzair, do you have anything? You joined us, I think, by the end.
I'm sorry. I think I really missed because of some time changes here in Canada. I really missed it, unfortunately. But I think I'm really grateful. And I had an opportunity to meet personally Dr. Faisal Khan during my recent visit to Pakistan. And we did discuss, and I tried to help him solve some of his problems in his lab. And I think that solution is working. Now they are very active and they are taking it to the next level. I'm so happy, really. And I was really impressed by his very, very passionate, keen interest, and moving straight from the Oxford to Pakistan, and to create his own pathway. So that's very impressive. A lot of best wishes and compliment to Dr. Faisal Khan.
I entirely agree. And
Thank you, Ji, and thank you for the introduction to APPNA Ji.
Very, very nice work overall, indeed. And definitely here in Stockholm as well, we are very happy. If you need any kind of help or somewhere, any collaborations from Karolinska Institute, Stockholm, please do contact us. We will see whatever we can come up with help or anything else. And with this, I think we will end today's session. It was very exciting and nice to see this fantastic work and labs run by Dr. Faisal. And finally, I will thank all the participants and also the panelists and the audience, everyone. Thank you very much. Okay, bye-bye.
Thank you, Ji.
Thank you.